This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 196, recorded on August 17th, 2012. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Got a big crew today joining me here in the TWiV studio from verticalfarm.com. Dickson Despommier.
Hello, Vincent.
Welcome back.
Thank you, sir.
Did you notice I said you were from verticalfarm.com?
I heard that. I did hear that. I appreciate the plug.
Plug, yeah. You want people going to those vertical
Absolutely.
farms. How's everything with you? Good?
Perfect.
Nice day today here, right?
It's a beauty. They even fixed our air conditioner here at work.
It is tolerable, too.
It was 3 weeks in the process of trying to do that. That was arduous.
Also joining us today, where should I start? From Western
Alphabetical.
Massachusetts. No, it's too hard. Alan Dove.
Good to be here.
How are you doing, Alan?
Doing okay. So it sounds like the facilities maintenance in the Hammer building hasn't changed a bit.
That's a fact.
Oh, it was.
That is a fact.
The AC was out for a month.
Yeah, now it's too cold in most of the labs.
Here outside, it's 29 degrees Celsius.
29.
Yep, it's about the same here.
Same there.
Right.
Nice day. Also joining us from North Central Florida, Rich Condit.
Hi, everybody.
How's it
How you
down
doing?
there?
Hey, Rich.
Is,
74 degrees. Get this. 74 degrees.
Really?
That's 23 Celsius.
Wow.
cool
raining.
there.
Raining. You could use it.
Yep.
Is that so? You're dry down
they
there
had
as well?
They had a drought. Big drought.
Yeah. Oh, yeah. It's serious.
All right.
Yeah.
Continuing here on this massive TWiV from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
I bet it's dry there, right?
It is dry here, but it's just about a perfect day. We also have 23 Celsius.
Nice.
All right, now we get into our guests. We have two guests today, both at the same institution over at the University of California, San Francisco. The first is Mark. Sorry if I mispronounced your name. Stenglein. Did I get that right?
You got it right.
Good. Mark Stenglein, welcome to TWiV.
Thank you. It's an honor to be here.
I understand you listen now and then.
I do. I listen religiously, so to speak.
Excellent. I'll tell everybody why you're here in a moment.
You should have said, "Bless you, my son."
Right, bless you. And finally, Mark's boss over at UCSF, Joe DeRisi. Welcome.
Hey, everyone.
How are you? Have you heard of TWiV?
Great.
Have you heard of TWiV, Joe?
I have.
Very good. Because it is about viruses, which is what you like after all, right?
Definitely.
Yeah, we will eventually get to the viruses. Right now, we're still in the weather report section.
That's right.
Hey, how is the weather out in San Francisco anyway?
It's fantastic, actually.
Yeah. I bet it's 72 degrees, right?
No, not quite, but it's close.
All right, the reason we have a big group today, well, you don't need a reason on TWiV. But anyway, today we're going to talk about a paper that just came out this week from the DeRisi lab. It was released in MBio on Tuesday. It's called, let's see. Let me get it up here so I can get the title right.
Identification,
Good.
Characterization, and in vitro culture of highly divergent arenaviruses from boa constrictors and annulated tree boas, candidate etiological agents for snake inclusion body disease.
Thank you, Alan. So I saw this paper actually a couple of weeks ago. I got an advance copy through ASM. And so I thought it would be pretty cool to do. And on Tuesday it came out, so I put it on the TWiV agenda. And that's when I got an email from Mark. Now, Mark, do you mind if I read a bit of your letter here?
No, go for it.
So his letter is, I'm a postdoc in Joe DeRisi's lab at UCSF. I previously wrote to tell you that the true ambition of my postdoc was to get onto TWiV. I hope you don't mind that, Joe.
No, that's fine by me.
I remember that letter. Everybody knows. Everybody's heard that before, so it's too late.
I'm a huge fan of this show, and I would view this as the ultimate achievement in virology. Anyway, the paper we published about the discovery of arenaviruses in snakes with filovirus-like glycoproteins may be my best bet. And so he sent links to it and said, I'd be happy to come on. So I asked Mark to come on.
Tell those losers in Stockholm he wants nothing to do with them. It's all about getting...
There you go.
That's right.
And then now as well we have Joe. So we're going to talk about this wonderful paper here today. This is pretty cool. And I think this is probably the only paper we'll do today. I doubt we'll get to anything else. So before we do that, though, I don't know anything about either of you guys. Let's start with you, Mark. You're a postdoc in the lab. Where'd you come from before that?
So I started my academic career at Washington University in St. Louis as an undergraduate, and I actually didn't study any biology there. Instead, I was a math and French major. Right. And then I don't really have the typical scientific career. After
but math and French go really well together, right?
They do.
Hey, we, Come on. So after graduating from WashU, I worked for an engineering company for 6 years, actually, and liked some aspects of that job, but then decided I wanted to switch into a research career. And so I was back in Minnesota, where I'm originally from, working, and so I started taking classes at the University of Minnesota in biology. I got really interested in that, and then decided to go to grad school there. So I went to graduate school at the University of Minnesota and got a degree in biochemistry and biophysics from the University of Minnesota, and there I was studying antiviral immunity.
Whose lab were you in?
The lab of Reuben Harris.
Okay. There are a couple of other virologists out there in Minnesota, right?
Yeah, there's actually a number of really excellent virologists there.
And the ASV was there last year. You didn't happen to go to that, did you?
I did, and I saw the TWiV live podcast there, which is cool.
And then you decided to do a postdoc with Joe?
Yeah, one thing that attracted me to Joe's lab was obviously virus discovery per se seemed like a really fun thing to do in science. And also, one really great thing about Joe's lab is the application of these cutting-edge genomics techniques, which I didn't do much during my PhD career. So I wanted to do more, sequencing and microarrays and bioinformatics, which is something that we do here. So that's. And then obviously Joe is a great mentor, as is Don Ganem, who. Originally, when I started the postdoc, I was co-mentored by Joe and Don. And Don has since left UCSF to be the global head of infectious disease research at Novartis. So now I'm fully in the DeRisi lab. But, the opportunity to work with such great mentors was really attractive.
How long have you been there now?
2.5 years.
So how old were you when you entered graduate school?
Let's see. Do some math here. I was 29.
Okay. That's relevant. We get questions frequently about, is it too late to change careers? Am I too old to do this or that? And your career path is an interesting one.
Yeah, so it can be done.
Mm-hmm.
Right. How about you, Joe? What's your background?
Right. My background's a little bit more linear than Mark's. I went to undergraduate at the University of California, Santa Cruz.
I was from Santa Cruz.
Really? What college?
What? I was Cal College.
Well, I was at Crown College.
Okay, what year?
So I was there from 88 to 92.
Okay. You're a baby. I graduated in 1970. I was in the second graduating class.
70.
Whoa! Yeah. You got me there.
Right. It was only 1,200 kids when I entered.
Wow.
It was great. Anyway, Santa Cruz, fantastic.
Yeah. So I got my bachelor's there in biochemistry and molecular biology, and from there I went. I took a lot of classes actually from Harry and worked with him a little bit.
Yeah, he was the first lab I was in. He got me on this path.
Yeah,
Terrific.
He definitely converted me down to go to graduate school. I actually was not going to go to graduate school and sort of bail out and, who knows, paint T-shirts at the boardwalk in Santa Cruz or something.
That's right.
As graduates from Santa Cruz are wont to do.
Right.
But Harry actually sat me down and said, you really ought to go to grad school. And so I applied and ended up going to Stanford, and I was in the lab of Patrick O. Brown and Pat Brown's lab at Stanford, where I wanted to be a virologist and initially started working on HIV and MLV integrase enzymology. Did that for a while, but then got sucked into developing genomics technology while in Pat's lab and ended up doing a lot of model organism yeast biology using DNA microarrays and building robots and all that kind of fun stuff. And from there, I was recruited directly to UCSF. So I took a fellow's position at UCSF for a year and then joined the faculty.
Did you overlap in the Brown lab with Ela Singh?
I did. In fact, she taught me a lot of cell culture techniques for handling MLV and purifying retroviruses and all that. You bet.
Yeah, she was here for a while. And I remember when she came here, she brought some technology from the Brown lab to her lab here at Columbia.
Yep.
All right. So let's move into this cool story. And I guess the first thing we should start with is maybe, I don't know who to ask, so you guys can decide who to answer this one. Maybe you should tell us a little bit about this snake disease, inclusion body disease of snakes, because I'd never heard of it before.
Hey, Mark, why don't I start with the initial letter?
Yeah, that's a good idea.
So I hadn't heard about this disease either, and I doubt Mark had as well. Snake diseases are not at the top of my list on a daily basis. And we had just finished a body of work on avian bornavirus, which causes a disease, cause parrots and cockatiels. And after publishing that work, I had gotten a typewritten letter in the mail with a photograph. The photograph had a woman in it with a boa constrictor sort of wrapped around her neck.
In a friendly way, I hope.
In a friendly way.
Yes,
yeah, in a friendly way. And the letter said, I've come to know of your work, and I want you to know that in boa constrictors and pythons, there's a disease that's been plaguing those snakes, especially domestic owners in aquariums and zoos, called inclusion body disease. Actually, it said IBD at first. And I thought, that's weird. Do snakes get inflammatory bowel disease? But it means inclusion body disease. And she went on in the letter to say, look, my snake, Larry, is very important to me. And it turns out that Larry does not have IBD, but she previously had snakes that died of IBD. And so she's very concerned about this disease, inclusion body disease. And she sort of asked me if I would work on this disease. And I, frankly, put the letter on the desk. It got buried by a bunch of other papers and sort of forgotten for a while until I was sort of cleaning off the desk as I want to do when procrastinating on writing a grant. I was about to throw away the letter, but then something about the picture caught my eye, and also the veterinarian's name and number that she sees was at the bottom of the letter. And so I thought, what the heck? I'll call the vet up and ask about this IBD thing. I called the vet up, a guy named Chris Sanders, and he said, yeah, IBD is a real deal. It's probably one of the most worrisome diseases we have to think about as veterinarians dealing with snakes. And zoos and large collections are constantly threatened by this. And it's literally worldwide. It's been around for decades, and no one's cracked it. Even though you can see in the microscope, very obviously, large cytoplasmic inclusions. And that
smells like a virus, yeah.
Oh, it screams virus, right? And so a little bit more investigation on the topic revealed that there wasn't that much work going on in the area. People had made some tries to sort of proteomically characterize the inclusions and so on, but hadn't really made a lot of progress. And so that's basically when we decided to do it.
There's not a whole lot of funding for studying snake diseases.
Yeah, there's a bigger generality there. There's not a lot of funding for veterinary virology, period.
Right.
In anything.
Right.
And perhaps outside of boiler chickens and cattle, right?
Yes.
And so, it's sort of an interesting area of virology that is, I would say, a little bit neglected. And so I am very enthusiastic about getting involved in projects in this area, simply because there hasn't been the funding to do there, and we have a little discretionary funding that we can go after some of these things with.
Right, and in terms of relevance, it strikes me that zoonotic infections come usually not from broiler chickens and cattle. So maybe we should be a little more worried about these, right?
Well, I mean, you've got West Nile virus as a good example of a recent outbreak that's veterinary-based that spills over into people.
Yeah, absolutely. I think the chance, who knows what the chances of any of these things are of moving from one species to the other, but we know it does happen, and there's a historical precedent for it. So, yeah, we should be concerned about it. And oftentimes, these vets are incredible resources. They have a lot of knowledge. They've seen a lot of the diseases. They know what the phenomenology is, as well as the epidemiology. And they don't have any money to work on this stuff.
So is this a disease of captive snakes, or do we know that it occurs in the wild? I guess that would be hard to find out, right?
You're absolutely right. I don't think anyone knows anything about what it is in the wild. There's a couple reports of wild-caught snakes with the disease. However, they had been in captivity for some time before they were assayed, so it's not clear where they got it.
What's the mortality rate from this?
It's essentially 100% fatal.
Wow.
Now, there's a little wiggle room there because older snakes can linger quite a long time with the disease. Younger snakes appear to succumb quite quickly, but older snakes can sort of, limp along with the disease.
Right.
And it's also thought that some species of snakes progress faster than others and so on. But, that's, of course, based on sort of loose data.
Just constrictors or vipers also?
And there's no evidence that I have read or seen in all the vets we've talked to that it affects vipers or poisonous snakes, that this really is a disease of the constricting snakes.
Which in itself is really interesting.
Very, especially Rich, considering how many of those released Burmese pythons are running around.
We have sort of a surplus of pythons down here. I've been looking into this, but we can get to that.
Yeah, that's a burner number 2.
And I'll just lead off with one more thing before I pass the baton to Mark here. So, we didn't have snakes in the lab to exactly work on this or anything, as you can imagine. And,
right.
and so the route that we took was actually contacting local vets and pathologists. And one of the local pathologists who looks at, slides of these diseases and things for vets around the country and around the state, a woman named DeRisi, reported that the California Academy of Sciences, right in our backyard, was sending her quite a number of samples for IBD diagnosis. And that, this is evidence that they were having a problem with it. And that we should get in contact with those guys. We sent a letter to the vet, Freeland Dunker, over at the California Academy at the Steinhardt Aquarium, where they have a wonderful collection of reptiles. And sure enough, they had a room that contained annulated tree boas and boa constrictors. And they suspected they were having a problem. There was a young snake that died very early. We unfortunately don't have tissues from that one early snake. But on the basis of the symptoms of that snake and diagnosis of IBD in that young snake, which they called a poor doer, as in not doing very well, they sent out biopsies from numerous other snakes in the room, and those biopsies were coming back positive. That's when we got involved because the tissues were then available. Do you want to take it from there, Mark?
Sure. Yes. So
he's mesmerized.
Yeah. So the starting point of our investigation was these samples from the snakes from the Cal Academy, like Joe was describing.
So, Mark, before you tell us, did Joe come in the lab and say, "Hey, Mark, you want to do this?" Or did he throw it up in the air and you caught it, or what? How did that work?
So actually, the project was started before I joined the lab, and the initial investigations were done on by another postdoc named Amy Kistler, who's an author on the paper. And what she did was, and this is sort of an interesting commentary on how fast the technology that we're using is changing to do these kinds of investigations. Because when Amy, so this is just back in 2009, when Amy first looked at these studies, she used what's called the virochip microarray to look for viruses in the samples from the IBD cases. And as you may or may not know, the virochip, it was developed in Joe's lab and was a viral detection microarray. And the virochip is based on hybridization of viral nucleic acids in your disease sample to virus sequences that are spotted onto a glass slide, onto a microarray. So the viral, in order to be successful, the viral chip requires there to be a certain amount of nucleotide similarity between the sequence of the virus in your sample and the sequence, and a viral sequence is already on the microarray. So Amy actually got the samples and attempted to use the viral chip to identify viruses in the samples, but it didn't work. And now that we know what the viruses are, it makes sense why it didn't work, because they're just too divergent to be detected by that method.
So this project was started back in 2009?
Right.
So I think that's important in particular for our listeners, because even now with some experience, anytime something like this comes up, it feels to me like, oh, this just happened yesterday, right? But you guys have been thinking about this now for 3 years, at least.
Yeah.
It takes a while.
So veterinary pathologists have known about these inclusions for a long time. Why hasn't the electron microscopy revealed the virus?
Well, that's a really good question. There's a ton of EM work, in fact, on inclusion body disease. And you can see large aggregates of protein. You can see the inclusions. But virus-like particles are hard to see. They have been seen, but it's just on the basis of what a virus-like particle looks like.
Right.
An identification was not possible.
Got it.
False starts were made. So in one attempt, one investigator thought that it might be a retrovirus based on the VLPs and was attempting to purify the entity based on transcription activity, and efforts along those lines. So I think you can see particles, you can see the aggregates. However, it was not sufficiently discriminatory enough to nail it for the viral species that it was.
So, Mark, what kind of samples did Amy and you get from the academy?
So we got samples from 6 different snakes, 5 of whom were confirmed as IBD positive by the pathologist, and 1 of which was negative, but they're all housed together. And from these snakes, so they were diagnosed while they were still alive, but then because they were diagnosed as having IBD, they needed to be euthanized just to prevent the spread of the disease to the rest of the aquarium. And so at necropsy, Amy collected heart, lung, liver, kidney, GI tissue, and those are the samples that we worked with.
So this is that's something we should come back to later. So it's known to be transmissible in a place like an aquarium or wherever snakes are kept, right?
Yeah, there's actually published transmission experiments.
Okay. So
is it respiratory, or is it ingested, or?
The route of transmission, the natural route of transmission is unknown. The experiments, the inoculations were, I can't remember how the inoculations were done, but the sort of natural route of transmission is unknown.
But do you have to have snakes in the same cage to have transmission? Is that known, or?
That's not known. There's just not been that detailed research done on this disease.
We should probably try this out in ferrets.
I was thinking the same thing.
Yes.
They're such a nice model for boa constrictors.
Yeah, really. So what did you do that was different that got you somewhere?
Yeah, so about last year, I actually picked up this project that we decided would be interesting to apply the newer technology that we're using to look for viruses, which is deep sequencing-based approaches to virus discovery. And so basically what this involves is taking nucleic acid from the samples and basically randomly sequencing everything in the sample.
Wow.
And the idea is that if a sample is has a viral infection, we should be able to detect the viral nucleic acid in the sample by this random sequencing approach. And this technology is evolving at just a breakneck rate, and now we can routinely get tens of millions of samples or tens of millions of sequences per sample at a reasonable cost and reasonably easy.
So you took all the tissues that I presume you had nucleic acid and subjected them to deep sequencing.
Yeah. So what we did, I took RNA actually from all of the tissues and deep sequenced it. And then what I had was hundreds of millions of sequences. And obviously the next challenge is, okay, what do you do with 100 million sequences?
Exactly.
And that's where the bioinformatics comes in. And one thing that turned out to be really helpful for this bioinformatics approach was to be able to filter out the snake sequence. Because obviously, if you take snake tissue and you sequence it, most of your sequences are snake. And so until very recently, there wasn't a boa constrictor genome sequence. But luckily, Joe had a ingenious idea that maybe he can explain.
Right. So, obviously the problem is if there's no boa constrictor genome and there are endogenous viruses in the genome, and how would you know what was exogenous or endogenous and so on? So it's kind of a real challenge. It's about this time that actually I was on an advisory committee with David Hausler and Ian Korf at UC Davis and UC Santa Cruz talking about a competition, a collaborative competition in which groups would get together and try to assemble genomes from the random bits of sequence that come out of these deep sequencers. Because the assembly problem of putting together genomes is kind of still a hard bioinformatics problem. And so the idea was, let's take a species that's never been sequenced before, let everybody try to assemble it, and we'll compare methods and see what works. Well, when it came time to figure out what species that would be, I suggested, hey, why not the red-tailed boa?
Just as a random thought.
Just off the top of my head.
That's right.
Off the top of my head. Now, there was actually really good reason to do that. There had been at that time, no snakes had been sequenced. So you couldn't rely on a closely related species as a crutch to help your assembly method. It would be a true test. And Illumina, the company, kindly agreed to sequence all the stuff for free.
Wow.
And they sequenced the boa, and then all these different groups, including the Broad and BGI and all these other genomics groups, competed in a friendly way to put it together. In fact, we here at UCSF tried to do it ourselves, and we didn't do very well, but we did sufficiently decently enough to have a submission to the competition. The end result was a pretty nicely assembled boa constrictor genome. And that's exactly what Mark needed to sort of filter through the haystack looking for the needle.
Wow.
So this is this Assemblathon. There's a website for this, right?
You bet. This is Assemblathon. Now, there's two Assemblathons. There's Assemblathon 1, which was a synthetic genome, kind of a fake genome, right? Which, that's not as much fun. Assemblathon 2 is the one we're talking about here, where real data with real species were used. And one of the species, it wasn't the only one, there was a fish as well, was the red-tailed boa. So Assemblathon 2. And anybody who wants to go look at that data can actually go to the website and download it.
So, Mark, did you do the filtering yourself, or do you guys have a group that does that in the lab?
No, I did it. And one nice thing about working in the DeRisi lab is that we're encouraged and allowed to do both the so-called wet bench science. We were actually pipetting and working with samples, and the bioinformatics. So there's a lot of there are some people who specialize more, but most people do both the wet bench and then the bioinformatics, which has been a great training opportunity for me.
Well, especially if you're going to go off and have your own lab, then at least you know how to do everything, right?
Right.
That's always a problem I have in big labs where everything is divided up. No one person gets to know what they need to go forward.
Right.
Yeah. So how long does it take? Give us an idea. You're starting with these millions of sequences and you're subtracting out the snake. How long does it take to do?
That probably took about a week of computers, and we have, these are big computers with lots of memory and lots of processors running 24 hours a day, and it probably took about a week.
And then the result is what is left after you subtract the snakes. So what do you do with that?
Right. So then that's a more tractable starting point. And like Joe said, most of the endogenous retroviruses and endogenous viruses have probably been removed, which reduces your sort of confusing false positives. And so with what remains, I searched against a database of viral protein sequences. And then immediately there was sort of an obvious, potentially interesting hit that came out of that.
So I had a question about that because in the paper you talked about using the NCBI non-redundant nucleotide and protein parentheses NR databases. And I just wondered if you could explain a little bit about what are the features of the non-redundant databases? Does that save computing time, or what does that mean?
Yeah, so the NR, the non-redundant database, the NR and the NT database are it's basically all of the sequences in GenBank, the nucleotide and all of the protein sequences in separate databases. And the non-redundant just means that if there's identical sequences, they remove them, or they collapse them, just to avoid. It just reduces the amount of time it takes to search through them.
Okay.
So you said you got a hit right away.
Mm-hmm.
It was obvious that there were multiple hits of the same virus.
Yeah, so there was hundreds or thousands of sequences that looked like arenavirus protein sequences, which is, but they were. You can tell that they were only slightly similar, only about 20% similar at the protein level, at the amino acid level, to previously described arenaviruses. Mm-hmm. So this looked like, it looked like it might be in a possibly new arenavirus. And I should say there was also sequences that were matching to filovirus protein sequences. Ebola virus and Marburg virus are filoviruses. And so it looked like there was at least one new virus in the samples.
So what do you do next?
Yeah, so next I was actually aided by a tool that was developed also in the DeRisi lab by Graham Ruby, who's another postdoc, and that's called the Price Assembler, which is a assembler that was designed specifically to solve the problem that I was facing, which is you have all of these sequences. A subset of them are what you're interested in. How can you try to combine all of those small 100 nucleotide sequences into a longer sequence? So it's the analogy that we like to use is that it's like what you're trying to do is put together a jigsaw puzzle, which in the analogy is assemble a large virus genome. But you have a box. So say you're trying to assemble a jigsaw puzzle with 1,000 pieces, but you have a box full of 1,000,000 puzzle pieces, and only 1,000 of them are from the puzzle that you're interested in. This Price Assembler is a way to do a targeted assembly where you basically are what you can do is focus on the pieces that you're interested in among all of the other irrelevant data.
So there could also be, so let's say there's your virus that you're interested in, but there could be other viruses in there. Will the assembler deal with that? Will it assemble multiple different genomes?
It can, depending on how you run it, yes.
Okay. So you ran that, and what came out?
So what we got out was apparently 2 viruses that looked like they were segregating with the 2 different species of snakes, the boa constrictors and the annulated tree boas. And what I essentially got out was the complete genome sequence of these 2 viruses.
So this must have been pretty exciting, huh?
Oh, yeah, that was very exciting. And I remember Joe was walking by my desk one day and he said, "What's going on?" or anything cool, anything new?
Just discovered 2 new viruses.
That's so great. That rare occasion when your mentor walks by and you actually do have some.
Yeah,
that's right.
Rare is the operative word here.
Yeah, for sure.
Yeah,
cool.
So you showed him this assembled genome of these 2 viruses. What did he say?
Joe, what'd you say?
I don't remember my exact words. And it's probably not appropriate for the radio podcast, but something in fact is, that is really cool.
So then, what did you do with these sequences, Mark?
So, sort of a lot of follow-up work, like confirming the sequence of the genomes using traditional Sanger sequencing and using RACE, or rapid amplification of cDNA ends, to confirm that we had the correct ends of the genomes, and doing things like quantitative PCR to determine the levels of the different viruses in the different samples. And then one of the other first things that we did was start the production of an antibody against the viral protein, which we realized immediately would be a tool that we would need to nail or to further study the virus and to try to investigate it in relationship to the disease that we proposed that it might be related to.
I just wanted to insert that when I was reading this paper, I made a note that I thought it was a really nice rationale for making the antibody to this particular protein. So I don't know if you want to go into that a little bit, but I thought that was really convincing.
Yeah, so we made an antibody in rabbits against a peptide from the viral nucleoprotein. And there was a few reasons for doing that. One is that it's the most antigenic in other arenaviruses. This protein is the most antigenic and the most abundant of the viral proteins. And also its predicted size was 67 kilodaltons, which was quite similar to the size of a protein that had been purified biochemically from inclusion body tissues before as the predominant specific protein species in those samples. So we thought maybe this is the inclusion protein.
Do other arenaviruses make inclusion bodies?
They do. I don't know if they make precisely the same type of inclusions, but they form cytoplasmic aggregates.
In fact, LCMV is another arenavirus, and the cytoplasmic inclusions that have been previously published look almost identical, at least by immunofluorescence, to the kind of things that we ultimately saw there.
Okay, and so is it known that those things are contain a lot of nuclear protein?
Yeah, almost exclusively containing NP.
Right,
nuclear
so
protein.
that makes sense. Okay.
So what do you do? Sorry, go ahead.
I was going to make one point here. This is an interesting thing. Previous approaches were to purify the inclusion protein. So if you purified the nucleoprotein from these inclusions and then try to do mass spec proteomics and sequence it and try and figure out what it is, remember, these proteins are only less than 20% identical to anything out there. In which case, your chance of finding it, first of all, it's not going to be in any of the databases. So if you try to match up your mass spectra with the database, you're going to get 0. And then if you try to do Edmond degradation and sequence the protein, you're only going to get a handful of amino acids. And so your ability to actually pick it up as an arenavirus for what it is through a protein purification approach is, that's going to be rough going.
Yeah. So what did you do with this antibody, Mark?
So the first thing we did was confirm on a Western blot that it reacted to what we thought, a protein of the predicted size, and then we used it to do immunofluorescence microscopy on cells that were infected in culture and infected tissues, and we're able to show.
That's an important point, Mark.
What's that?
The cells. I mean, you should actually speak about the cells.
What
themselves.
kind of cells did you use?
Yeah, so that is a good point. So. We, in addition to an antibody being an important reagent to study this virus, we realized that isolating the virus in culture was going to be important for a number of reasons, including the fact that if we were proposing that this virus caused this disease, to demonstrate that, what you really need to do is to infect snakes.
Right.
And to do that, you need a source of the virus, and a convenient and good source of the virus is to use tissue culture. Virus that you've replicated in tissue culture, and you can make stocks of the virus that you can use for these experiments. And also, having a cell culture replication system enables you to do a lot of sort of basic virology, basic biology. And so I tried using homogenate. Basically, you take frozen tissue from the infected snakes, you chop it up and grind it up, and filter it, and you put it on cells growing in culture. And I tried infecting so when we first started, we were like, okay, what cells can we try to infect? And so we looked at the ATCC, which is a repository of cell lines. And there was 4 reptile cell lines in the ATCC. There was a turtle cell line, an iguana cell line, and 2 viper cell lines. And so we tried infecting those, and we could not get the virus to replicate in those cells. And we also tried to infect vero cells, which is a African green monkey cell line that has been used to replicate a lot of different of the previously described arenaviruses, but the virus would not replicate in those cells either. So that was a bit of a roadblock for us, and we weren't sure exactly if we were going to be able to get this to work, but we knew that one possible explanation for the failure of the virus to replicate in these cells was that there was a species barrier to replication, and what we needed was a boa constrictor cell line to grow or to replicate this virus that was infecting boa constrictors. And so conveniently around that time, our vet collaborator and friend Chris Sanders, and this was convenient for us, but it was a small personal tragedy for him. His personal boa constrictor, Juliet, was dying of cancer. And right around this time, Juliet actually died of lymphoma. And Chris knew that we were looking for boa constrictor tissue that we could use as a source to try to make cell lines. And so Juliet turned essentially into a boa constrictor organ donor. And I went down to Chris's vet clinic and necropsied Juliet with him and brought back a bunch of snake tissue on ice to the lab and did what you do to try to make a cell line, which is basically chop up the tissues and do some enzymatic digestion of the tissues and put them in culture, and then basically you just wait and see if any of the cells, because basically all of the cells immediately die. Almost all of the cells immediately just kick the butt when you put them into culture, kick the bucket, and not kick the butt. And luckily I tried a bunch of different tissues in a bunch of different conditions and got cells. After about 5 weeks, I got cells replicating, propagating in one of the cultures, which is a culture derived from the kidney, from Juliet's kidneys. So this turned into a continuous boa constrictor cell line that we named JK for Juliet kidney.
I was just going to ask, did you name them Jusa cells?
No, that would be good. Jusa.
So they are. You think they're permanent cell lines, immortalized, or not? You're not yet sure.
That's unclear. They've been growing now for 6 months. They've been propagating continuously for that long, but I think that does not necessarily mean they're immortal. I think they still could senesce at some point.
Yeah.
But
I'm sure you have some frozen down.
Oh yeah, we've got boxes and boxes full of tubes.
These seem to be growing in a fairly conventional medium. This CMEM, it's fetal bovine serum, just regular stuff. Is that right?
Yeah, I actually tried using chicken serum, thinking that reptiles and birds might be, being more closely related, might be better to use chicken serum. But actually, the cells died in chicken serum. So,
and 30 degrees. Did you play with the temperature?
No, I didn't. There was enough variables already.
All right.
So then you threw tissue extracts from diseased snakes onto these cells. Is that what was the inoculum?
Yep. So it's the same inoculum that I used on the other cell lines. It's basically a homogenate from liver and kidney from the Cal Academy boa constrictors, and put that on the cells. And sure enough, the virus appeared to be replicating in the boa constrictor cell line.
Now, how did you tell that? Because I remember in the paper you said there was no cytopathic effect, right?
Yeah, that's right. So, and arenaviruses generally are not known for producing cytopathic effects in culture. But I followed the viral replication by qRT-PCR. So I monitored viral RNA levels in the supernatant of the cells.
So if you take the supernatant and then put it on fresh cells, do you get another cycle of replication?
You do.
Because I don't remember seeing that in the paper.
That's right. It's not in the paper.
Okay. That's important, right?
Mm-hmm.
Because it shows that what you're making is infectious virus.
Yeah, that is important, especially if we're going to try to take the virus and put it into snakes.
Yeah. So can you take the cloned sequences and rescue viruses from those as well? Because now you have a cell line that you know is permissive.
In theory, you could. We haven't cloned the sequences into a vector that would allow that yet.
I presume
that's going to be a little problematic because you got to set up a whole rescue system, right, and express the proteins and stuff.
And that's been done for other arenaviruses. So in principle, it's possible, but we haven't done that yet.
It's like you need like 5 plasmids. It's like a 5 plasmid system to do it.
Right.
Yeah, that's why I started with a plus-stranded virus years ago. 1 plasmid.
Yeah, we haven't mentioned what the genome of this thing actually looks like, right?
Yeah, right.
It's 2 RNA segments, each of which is ambisense. That means it's single-stranded RNA, but it has each one has a gene. There are 2 genes each, and each one of the genes is in one orientation in terms of coding, and the other is in the complementary orientation on the same strand. Very weird.
Yeah, it is weird. My understanding of and this organization, although these viruses are quite divergent, the overall genome organization with other arenaviruses is extremely well conserved, which suggests to me that it's important. And my understanding of what the organization might accomplish is regulate the timing in some way of the expression of the viral genes upon infection of the new cell.
So these viruses, and there are two of them, right, from the two different kinds of snakes. These form a very distinct clade within the arenaviruses, distinct from the new and the old world arenaviruses. What are your thoughts about that?
Well, I guess my thoughts are that this is a separate clade or a separate group of arenaviruses, and it apparently diverged from the common ancestor of what we call the rodent arenaviruses a really long time ago, because people have suggested that the old and new world arenaviruses diverged from each other something like 35 million years ago. So, which means that these viruses have been around for at least that long, infecting snakes or something else.
So that's my other question. Do you think they're in snakes, or are they constantly going into snakes from, say, rodents?
I think that's a really excellent question, and one we don't really have an answer for yet.
So are you planning to go out and trap snakes and look and see if you find it in them?
Yeah. You want to talk about that, Joe?
Yeah, I think it's an interesting possibility. We don't know anything about the genetic flow of information here. It could be snakes impart arenaviruses to rodents and vice versa. It's not necessarily that they just eat an infected rodent. They could actually bite one, right, and it gets away, or who knows how it happens, but at some rate, some low probability you get species transfer. So one of the things we'd like to do is, first of all, look at these large collection of frozen reptile tissues that these various sort of biological repositories has. UC Berkeley has a very large collection, for example. That's one way to do it. There's field biologists that collect tissue samples. And then also we'd like to layer on the rodent map on top of that. And there's several ongoing projects around the country to catch wild rodents in various parts and look at the viruses they carry as well. So I think by overlaying those two pieces of information, we might have a better picture.
So this virus could be in rodents, for example. We just haven't found it yet.
It's a formal possibility. You know, I do think that there's probably going to be a big species barrier between what we found in boas, but however, I think that the discovery of snake arenavirus points to potentially a much larger diversity of arenaviruses in the reptile population than currently we're just looking at in boa constrictors.
Right.
We didn't realize how big the arena was.
Good, Alan. Thank you.
Well done.
It's a good title.
So I'm betting that you're going to find these arenaviruses in snakes and maybe other reptiles, and that it's no coincidence that you've got a reptile, which in my mind is a very old species, with a very old virus in it. It probably goes way back.
Yes.
It's on the tip of my tongue.
Not a forked tongue, I hope.
No. One more. Yes. The other issue that we haven't addressed here is the unusual glycoprotein of your arena, Mark. So tell us about that.
Yeah, so if you remember, I was saying that when we first got the hits back from when I was first examining the sequences that we had, some of the sequences looked like arenavirus proteins, and some of them like filovirus proteins like Ebola and Marburg. And once we had fully assembled the genomes, we realized that so arenaviruses have four genes, and one of them encodes the viral glycoprotein. And what we found was that the viral glycoprotein had no detectable similarity to arenavirus glycoproteins, even though it was in the same genomic position. But instead, it was most similar to the glycoprotein of filoviruses like Ebola virus and to avian retroviruses. And so it appears that this virus is recombinant, some combination, or perhaps it's actually the ancestral configuration of arenaviruses. But it's sort of a mashup or a mix of arenavirus and filovirus genes.
It's going to be really interesting to find the receptor for this, because you probably know there have been some really interesting filovirus receptors identified lately, and I wonder if this will be in the same family or something totally different.
Yeah, I mean, that's a really interesting question, and I'd say it's one of many that we're interested in continuing to follow up.
So there are constrictors found on almost all the continents, plus Australia. Have you noticed any inclusion body diseases in pythons and constrictors from Australia? that might show a geographic isolation and perhaps some more unusual features that didn't come out right away?
That's another interesting question, and there are published reports of IBD in a variety of snake species, but it's not. I mean, it's best characterized in I think most observed in these boa constrictor species, and that might be a sample bias because those are the more common.
And where are they from? I'm sorry, I meant to ask you where they're from also.
Which
ones that you've isolated your virus from?
The snakes?
Yeah.
Well, the snakes are from the local museum. I'm not sure where they came from originally.
Okay, so they're
boa constrictors in general are from Central America, South and Central America. So that's if we can assume that the virus infects those snakes in the wild, that's where the virus would be.
I see.
So just a comment on this arenavirus, filovirus thing. The filoviruses are single-stranded RNA negative sense viruses. So they don't have a segmented genome like the arenaviruses. They're also membrane. Both the arenaviruses and the filoviruses are membraned. So they don't have a segmented genome, and it's not ambisense, but there are a lot of other, even though they look superficially in terms of genome organization different, there are a lot of similarities. Like they got this huge polymerase, as many of the negative strand viruses do, that's a multifunctional enzyme, and a nucleocapsid protein and a glycoprotein. So it's not a completely bizarre idea that there's some very ancient ancestor that has from which both the arenaviruses and the filoviruses diverged.
Yeah. I think it's worth pointing out here that previous to this discovery, Galler and Buckmeyer published a paper some number of years ago on the basis of gazing at secondary structure in glycoproteins between arena and philo, and made the hypothesis that they did in fact share a common ancestor based on those similarities.
So how's Larry?
Larry's
actually doing fine. You know, he doesn't have IBD, which is sort of the irony of this whole thing.
So the snake turned out to be a red herring.
Well, Larry doesn't have IBD, but you know the owner was rightfully scared of it because she had lost snakes prior to this due to IBD.
The snake turned out to be a red herring. I like that.
And I noticed that you, in your acknowledgments, you thank both the snake's owner and the snakes, which is nice.
Yep. That's right.
I think it's funny that this whole thing got started by a snake that doesn't even have the disease.
She doesn't happen to be an ecdysis by any chance, does she? Because if she was, then she should have an ecdysis partner, which is the snake, of course.
Yeah.
But
is that a joke?
I was just looking for a connection here as to why she seems to want to collect snakes. Or why anybody would want to do that. In fact, I've known kids that collect snakes, and then they lose their interest and they release them in the wild, and that's how they get distributed.
Come on, Dickson, you collect parasites.
Not on
weirder
purpose.
things out there.
Not as pets. So here, this is something that I want you guys to talk a little bit about. There's all kinds of interesting virology to do here, but you know, NIH is it's getting really tough to do this kind of work unless it's got some kind of human health benefit. And I know we've talked about the zoonosis angle, but you know, it sounds like you're going to get into it way beyond that. So how do you justify doing this? I mean, not to me, because I think it's all great to do, but in today's climate, how do you do that?
Well, I'll make a comment on that, Mark, and then maybe you can weigh in too from your own perspective. You know, if I was a young investigator starting out trying to write an NIH R01, you're right. I think it would be hard to do in this climate that's sort of driven towards translational human biomedical research. And I think it might be hard to get by some of the review boards now. However, one might consider alternative paths like going to NSF. Or other funding organizations. I've been fortunate enough to have, you know, the kind of discretionary funding through Howard Hughes Medical Institute that I can kind of have a little bit more freedom in what I get to do. Yeah. But I see your point, and you know, I think it's a big problem that real traditional basic science is taking a bit of a back seat to what is viewed as purely biomedical human biology research, because we know that the stuff that you find in basic research can have real implications for humans that are totally unexpected.
Yeah.
So, I mean, if you don't mind me jumping in here, because the only area that I feel competent in talking about is ecological stuff right now. So, what if you were to show a connection between these pet owner infections and the real world, in the wild? So, boa constrictors, they play a real role in rodent control and in keeping down a number of other pests, I'm sure, animal pests of sorts. So what happens if all the boa constrictors were to disappear? You could make a case that you don't want that to happen, just like all the amphibians are disappearing right now for something other than this, of course. But so NSF would be a perfect place, but also ecosystem balance and management and biodiversity, all that fits together. So you could make some kind of an ecological case, at least, for wanting to know more about this.
Yeah, I absolutely agree with that. And I think NSF would be the right kind of organization that would support this kind of research. And so, my advice to young people getting into, let's just say what we're doing here, which is really basic virology, is, have an open mind about the funding mechanisms. NIH is not the only game in town.
So, Mark, do you plan to take this with you at some point? And if so, how are you going to deal with this?
That's an excellent question. I guess the part of that answer depends on what am I going to do next, which I don't know if I've fully worked out in my head. I'm sort of in the process of trying to figure that out now. And I think it's really, like you said, there's a million different ways this could go. And being able to work on it would, obviously depend on finding funding for that. And as we've just been discussing, it's probably not going to come from the NIH, but there may be some other opportunities.
Joe, do you plan to continue this for the foreseeable future, this line of work?
Yes, definitely. I think the next big step is actually looking at some of those field studies and some of those broader reptile and rodent surveys. Again, that's stuff that would be hard to get funded any other way right now as well. Is just sort of this animal surveillance. However, I think that there's a lot, there's way more to do than any one lab can do. And, I would hope that Mark is able to take a big chunk of this virology and go to town with it and really set up shop. Now, one thing that is comes out of this is that, again, we talked about how veterinary virology is a bit of a neglected field. I really think there's a huge opportunity there, and evidence of the success that we've had, starting with the avian bornavirus and now this one and so on, points to a whole collection of diseases and viruses that have not gone discovered and yet may have importance in human health, and therefore should be fundable.
Well, I agree. I think the veterinary angle has been brought up before, and we have to try and engage them in doing more of this. And they have a little presence at ASV, American Society for Virology, but probably it needs to be bigger than that as well.
Right.
So, Mark, how much longer you plan on being around there?
Oh, that's a good question.
Joe, how much longer do you plan on keeping Mark around?
I haven't been ready to be kicked out yet, so that's good. I still have a desk.
No, we want him to get a job offer.
There you go.
So, and I've been doing some investigations in that direction and talking to some departments, but there's nothing concrete yet. So
you'd make a great
and maybe you'll get the TWiV bump.
Yeah, that'd be great.
You'd like to stay in academia?
Yeah, I think so. I mean, sometimes when I look at people I know or young PIs, young lab heads, they all seem extremely stressed out. And so I sometimes ask myself, is this really the road that I want to go down? But on the other hand, I love doing science, and it seems like that's sort of one of the greatest ways to do science. And so I'm having a little bit of an internal struggle, actually.
Think about the World Wildlife Fund, for instance. They could use people with your knowledge to help them identify problems. And so I think that'd be great. Peter Daszak would be a good person to get in touch with, I think.
Okay.
If you have the passion to do it, you should do it. That's really the determining factor, I think.
Yeah, I agree. And I love science. It's hard to imagine doing anything else. So
I know what you mean. And I think everybody on this call knows the same thing, right?
Yeah.
Yep.
Yep.
Anybody have anything else before we move on?
I just had one thing, and that is that I really appreciated in this paper, for instance, in Figure 1, where you show the histology, and you talk about the inclusion bodies, and you tell us that they appear orange and brown. When I read pathogenesis papers, or at least when I did in the past, and they'd say, and you can see this in Figure 1, and if you didn't know what color you were looking for, you were hard-pressed to figure it out. And you've pointed everything out with the white arrows and stuff. And then what I really liked, we kind of touched on this a little bit, is that in Figure 6, then you actually look at the inclusion bodies and you stain them with your antibody. So it kind of has come full circle that, you've identified what is probably the nucleoprotein in the inclusion bodies. And I just thought that was worth commenting on.
Yeah, thank you. That's Figure 6 is one of my favorite figures that I've ever made, and it really is a nice full circle where we show that the inclusions, which give the disease its name in our diagnostic for the disease, contain the viral protein, which we don't have formal evidence yet that these viruses cause the disease, but this is
right. I was going to ask, you have not put this cultured virus into snakes, is that correct?
Right. We're working on as you can imagine, doing experiments in snakes is a little bit of a logistics challenge. But we're working on getting that set up. We have our good collaborator Chris Sanders, the vet, is agreed to work with us on this, and we're now doing things like getting IACUC approval to do those studies.
You can imagine the phone call with the university when you call the animal facility and say, "Hey, would it be okay if we house a couple boa constrictors down in the mouse house?"
That's right. That's right.
Yeah, you need to collaborate with somebody with a vet school.
Yeah,
exactly.
I mean,
right.
dealing with reptiles here is not a problem. Okay?
Right.
We're lousy with them.
Exactly.
So, is there evidence for inclusion body-like diseases in other reptiles that you're aware of?
There are other viruses that when they infect reptiles, they cause inclusions, and so there are other viruses that are known to cause inclusions when they infect reptiles. Yes.
So, Rich, I can't believe you haven't asked the question though, because you got these Burmese pythons all over the place in South Florida.
Yeah, well, I've been investigating this. Okay, so this virus, my understanding is that this virus, or there is an inclusion body disease in pythons, put it that way. And so one of my first thoughts was, is this a way to biologically control the pythons that are taking over the other Everglades? And so I wrote some of my reptile buddies about this. And Elliot Jacobson, who we talked about before the show, who's and specializes in infectious diseases of reptiles here at the University of Florida, wrote me back and said, no, it's not going to work. Let me see if I can find his email here.
Well, there's a regionally appropriate solution to that whole Burmese python problem.
Yeah?
Yeah, them's good eating.
That's right. Tastes just like chicken.
You're in the South. I don't see why there's a problem with critters.
Tastes just like chicken.
So Elliot says, been asked this the other day. You can't use the virus to trash the Everglades pythons. He says, not at all. Probably the causative agent is the worst of pathogens to use because many snakes are subclinical and live for years while being infected. And later on, he says, plus, for some reason, it's rare to see a python with this disease. Burmese were commonly infected in the late 70s, but not anymore.
Huh.
Okay? So, that's not going to work down here, but...
Eat them.
Eat them.
Eat them.
My last question is, can you get these viruses to form plaques?
So, in the since they don't cause cytopathic effects in cells that we've observed, that may not be possible to do that traditional plaque assay. But we've thought about doing things like a fluorescent focus assay, which is sort of a fluorescent staining mediated plaque assay. So we have not got that up and running, but we've talked about it.
Yeah. So meanwhile, you're just quantifying by PCR, right?
That's right. Yeah.
For viral genomes.
Mm-hmm.
All right. We're going to move on to some email. Joe, if you want to leave, you can do so now, or you can stick around. It's up to you.
All right. Yeah, I've actually got a meeting, so it's been really great talking with you all. It's been a lot of fun. It's really fun to do one of these sort of high-level podcasts.
We enjoy it.
Great talking to you.
We're glad you came.
Thank you for using the word high level.
See you. See you, Joe.
See you now. Bye.
Bye-bye. All right, now, Mark, you can say whatever you want.
All right, true. All right, let me tell you guys the real story.
See, Joe's going to listen to this on the way home tonight, so I wouldn't do that if I were you.
He's not going to listen to it, no. You want to start from the top now?
That's right.
All right, let's do some email. Before we do that, I just want to tell everyone we are looking for a virologist here at Columbia.
Don't they already have some?
We only have 2 or 3.
Wow.
Me, Steve Goff, and a new fellow, Sagi Shapira. So we got an ad in the cell. And if you're a virologist looking for an academic position, we'll put the link in the show notes so you can check it out.
Mark,
What kind of virology will do?
Mark, they have pythons up at the Bronx Zoo, by the way.
All right.
It doesn't. It just says whose research interests lie in the areas of basic mechanisms of replication or pathogenesis.
Bingo.
Okay, great.
I like
that's
that
everything.
description.
Yeah, I like basic mechanisms of replication. That's becoming neglected.
You know, the snake story is interesting, Mark, because, look, the viruses are not cytopathic. So then how do they cause disease? Right. It's probably an immune thing, and that can get you into host pathogen interactions. It could be relevant anywhere. That's how I would spin it.
That's a good idea.
Anyway. Let's see. Who would like to take the first one? How about you, Alan Dove? Would you like to read this first email?
Dallas writes, I'm going through the old podcasts when I have time. Number 41 covered ISA, a salmonid virus with $1 billion class damages history. When someone mentioned vaccines development, a comment was made about how can you vaccinate salmon. Most salmon are already vaccinated for bacterial diseases using automatic machines where the salmon smolt are lined up, given a shot, and released. When you have hundreds of millions of fish to deal with, the machines are fast. To give you an idea of what has happened to antibiotic consumption over time in Norway salmon farms as vaccines for bacterial pathogens have developed, there's a similar graph showing the increase before vaccines, but I couldn't find it. And he sends along this graph that is actually quite dramatic. Where you've got, I guess the green, so the yellow line on this thing, which just absolutely it plummets. It's like a company going out of business type of graph from some cartoon. You know, they just thing drops off to nothing from 1987 to 1994, and that's the use of antibiotics. And then there's a set of columns on the graph illustrating the number of fish being produced, and that is just steadily climbing. And now you've got more and more fish being produced, and the antibiotic use is just done. So he continues, virus in the wild fish stock are also a significant issue, but as an old marine biologist once told me, nothing ever dies in the ocean. It's eaten alive before it's actually dead.
That's really a scary idea.
Yeah. Well,
I mean, it's true, I'm sure.
Yeah, although I know whales and large stuff can die, and then it lands in the deep ocean and gets scavenged.
It's an ecosystem.
It is. Okay, he continues, that makes wild virus hard to study, but the source in aquaculture net pens is from the wild. Don't believe all the nonsense being spread by the anti-aquaculture activists and the commercial fishermen who don't like competition. For example, the red pigments used in the diets are the same chemicals that make wild salmon red, and the same as found in red yeast or red algae sold in health food stores and found in peppermeal. The color-added label was politically added by the Alaska commercial fishermen who were getting economically killed by the quality control possible with farmed fish. No sitting around on the deck for hours before processing. Omega-3 fatty acids are a required dietary component in salmon feed, and they end up with these fats in their tissue just like wild salmon. One dinner of farmed salmon or high-fat wild salmon species equals 10 or so of those fish oil pills and tastes a lot better.
Hear, hear.
I still haven't run across an episode on shrimp viruses that are resulting in consumers paying an extra $1 billion plus per year in their shrimp costs to cover virus losses.
Does he mean us? We should do a podcast on shrimp
Yeah,
virus?
absolutely. White
Yeah, I
spot
guess so.
shrimp virus.
I just want to add something
Now, is that
here.
wait? White
Go
spot
ahead.
shrimp virus, is that in farmed shrimp primarily?
Yes, it shows up in farmed shrimp, but I think that there's some. I don't know. Well, we got to do an episode on it, so I got to bone up on it. But it is trashing the farmed shrimp big time.
Okay. Because the farmed shrimp are also trashing whole ecosystems, but that's another story.
I just read a recent statistic from the World Fisheries Association that said today the world eats 60% of its fish it's farmed. So 60% of all the fish that's eaten in the world is farmed fish. That's remarkable.
Is that good?
Well, that's because is less and less of the wild stocks.
Because we fished out the oceans.
Exactly.
But don't these farms put
the
waste
Japanese,
into the ocean?
yeah, of course they do.
problem.
The Japanese are now starting to farm tuna.
Mm-hmm.
That's an amazing thing.
Yeah.
That's a pelagic fish that requires huge amounts of ocean to live in, and they're farming them.
What does pelagic mean, Dickson?
Means that the open ocean, just the open ocean.
Oh, I'm pelagic.
You live in New Jersey.
Joke.
That's true. You're haploid also. No, that's another topic.
This fellow sounds like he knows about.
Yeah,
I think he's probably in fish farming.
Absolutely.
All right.
Studied it anyway.
Yeah.
Tell us who to get for the shrimp virus.
Right.
Dallas.
Absolutely.
Rich Condit, can you take the next one?
Sure. Oops. I just closed the page.
No, what's
No, I'm back. I get it. Sorry.
Sorry, yeah, it's from Sam.
Yeah. Okay. Here we go. Sam writes, Dear TWiV, thank you so much for all these brilliant podcasts, especially episode 184, Reforming Science. Crowdfunding for science is an interesting grassroots mechanism to obtain support for small projects. You're probably familiar with Petri dish already, but I wanted to highlight this project. And he provides a link here. Only 28 days to hit the funding target. Would be great if you could mention it on TWiV, Best Sam, and he says he doesn't have any conflicts of interest, so he's not on this website. But so Petri dish turns out is a crowdfunding website that I guess does specifically science projects. So you can put a description of your science project on this site and ask for people to contribute to it.
I like it.
And actually, now there's still 2 days left. Actually, it's going to be expired by the time this goes live.
Yeah.
And he's managed to. This project is it's a mosquito project, right?
Yeah.
Virus-resistant mosquitoes.
Virus-resistant mosquitoes. Yes, it has to do with trying to make them resistant through the saliva, right? Giving saliva proteins.
That'd be great.
He's managed to collect $996 from 12 different backers. He needs $5,000 minimum for funding. So he's got 2 days left. Wish him luck.
He's just going to miss the TWiV bump because.
Yeah.
Sorry.
Oh, well.
But people should check this out. He's a postdoc at Yale, actually.
Right.
And he writes that he presented this work at the ASV meeting. Cool. But Mark, maybe you ought to think about this for your to get some support downstream,
Yeah, that's a great idea.
People
I think,
with snakes would love it, right?
they would.
Yeah.
The only thing is when you see things like this and you know how expensive laboratory reagents are, it kind of makes you, a little.
Yeah. What are you going to do?
Not to be a
with
pessimist,
$1,000?
but you can spend $1,000 in 5 minutes.
It's true.
Yeah, 5,000 is not a lot of money.
But
it could be enough to get him going.
Yeah, and it's nice to. I think the crowdsourcing aspect is really cool.
The homepage for this site is really pretty cool, and they got a whole bunch of different projects here from all realms of science.
Yeah.
There's a GIS mapping of Pacific killer whales,
Oh yeah.
etc.
I think you got to get people's attention. There are a number of these sites for non-science projects, and some of them really take off. And people ask for $10,000, they end up getting $5 million.
Yeah.
You know, it really depends on what you're doing. So I'm sorry we only gave him 2 days, but maybe people...
He should have had a viral marketing campaign.
Oof.
Truly. Kathy, can you read the next?
Sure. Mark writes, "Hi, Vincent et al.
This is not you, Mark, right?
This is not me.
In case you haven't seen this yet," and he sends a link to something on Courthouse News, and he summarizes, Merck has known for a decade that its mumps vaccine is, quote, far less effective than it tells the government, and it falsified test results and sold millions of doses of, quote, questionable efficacy, flooding and monopolizing the market, a primary caregiver claims in a federal antitrust class action. Alabama-based Chatham Primary Care sued Merck on Monday, the week after the unsealing of a False Claims Act complaint to relators. Relators, okay, yes, filed in 2010. Those relators, Stephen Kraling and Joan Lachowski, were Merck virologists who claim in their unsealed complaint that they witnessed firsthand the improper testing and data falsification in which Merck engaged to artificially inflate the vaccine's efficacy findings. And then I guess Alan made a comment.
So I looked into this when I saw the letter come up in the queue. The Department of Justice and FDA already investigated this, rejected the claims, said this is BS. And so these court documents are part of a lawsuit that I gather is the plaintiffs trying to take the matter to civil court to cash in on it, even though the claims are already rejected. So it was already the False Claims Act that, they essentially took it to the federal government. It would have been a criminal case if this were true. And I suppose, if you're an anti-vaccine conspiracy theorist, you might say, well, the FDA is in bed with the drug company. But the Department of Justice, the FBI comes in to investigate something, their job is to prosecute. So my thinking is that if they didn't think that this was valid, it's probably a load.
Now, one of the things that is happening here is that we are beginning to realize that the mumps vaccine, in fact, has never been as effective as we hoped it would be. It was originally put out there, and the incidence of mumps absolutely crashed. And now we're, 40 years out, seeing occasional spikes of mumps epidemics. And it turns out that the first of all, that you do not necessarily acquire lifelong immunity even to a wild mumps infection, number one. And number two, that the vaccine, the best vaccine, is not as efficacious as was originally thought. And this reminds me of the flu story we did a while ago, where you can look at vaccine efficacy a couple of different ways. You can look at a serological response, but that doesn't necessarily correlate to protection. You can look at ELISA, that doesn't necessarily correlate to protection. So it's a lot more complicated than it might look superficially, and a lot of people are trying to sort this out.
Yeah, there are also multiple ways of defining protection.
Right.
I think most people would say that if you got a vaccine and it keeps you from getting serious complications of a disease, then that's an effective vaccine. But if you still got infected with the virus and could act as a carrier, then by some definitions, that would be an ineffective vaccine.
One of the interesting concepts that I find here is that it's possible that early during the mumps vaccination history, that one of the things that was happening was people were getting vaccinated and then they were getting boosted with circulating wild-type virus. And as the prevalence of wild-type virus decreased, that boost has gone away, and that might account for some of the spikes, some of the little outbreaks of mumps more recently. So this is a really complicated and interesting problem.
It is. And the other thing that is certainly going on is that vaccination rates right after the vaccine was introduced were, in countries that could afford to introduce it, the vaccination rate was near 100%. because this was a bad disease and everybody knew it was a disease you didn't want your kids getting, and so you got your kids vaccinated. It was a no-brainer. So there was certainly a degree of herd immunity that was going on there that was not quantifiable. And now that vaccination rates have been declining because of these anti-vaccine campaigns by people who don't know what they're talking about, we see these outbreaks of various diseases, including mumps, and that's what's kind of shined a spotlight on, well, how efficacious is this vaccine really? Mm-hmm. So if we only have 80 or 90% coverage of the population, can we still prevent mumps? And the answer is maybe not.
So there's a similar thing going on with pertussis now. There's a couple of outbreaks, particularly in the Northwest. And some people think the newer vaccine, the acellular vaccine, which was introduced not too long ago, may be less efficacious than the original one, which was a cellular one, which had too many side effects associated with it.
Right.
And so maybe you get these outbreaks now and then, which all are not attributable to failure to vaccinate.
Well, and also there's another parallel with the pertussis story, that we now know that the protection doesn't last all through your life.
Yeah.
So a lot of these outbreaks are undoubtedly being fed by people who were vaccinated as kids, and now they're in their 60s or whatever, and their immunity's waning a little bit, and now they're capable of carrying it to their grandchildren.
Yep.
So one of the things to me that's important here is that one may find out over time that a vaccine is not as efficacious as you initially thought it was. And that may be true. That doesn't necessarily mean that somebody is guilty of fraud.
Right.
Yeah. This means that you found out it wasn't as good as we thought it was.
Right.
And so then you work on correcting that.
Yeah. All right.
So English vocabulary lesson, because I didn't know what relators were. They are people who file lawsuits.
Yeah,
so
right. I had to look that one up too.
Spelled O-R or E-R.
A whistleblower. Come on, it's a whistleblower.
Right.
Well, no, just plaintiffs.
Plaintiffs.
Yeah.
But in fact, these were Merck workers, though, right?
Yeah, they were, apparently.
That's a whistleblower. In my book, that's a whistleblower.
All right, we have to do 2 more emails. Okay,
yes.
I want to do one more, but I see we have to do 2.
We have to do the one after it.
Let me do this one. Is from Richard. I just thought you should know, since I guess this is deliberate, that what you are doing is working. So far, you have got me to learn more about science, specifically biology and virology, than I ever thought I would, to the point of working my way up to Kevin Aham's molecular biology course on iTunes U. Of course, I had to start with basic chemistry so as to understand things like Schwarzschild radius, etc. From there, I went to basic biology as well as all your virology lectures. Then I took a break to grow some bacteria and phage, then to use the phage to create plaques. I wish I had saved the instructions, as it seems the phages have gone away. Pages?
I think
web
he means
pages.
web pages.
I thought he meant phages have gone away, though I am sure I can find them on the internet somewhere. Yeah, you can't find phages on the internet.
No.
Right now, I'm round 2/3 of the way through the molecular biology course, as mentioned above, 2/3 of the way. I'm sure that you will have had a similar effect on some small percentage of your listeners. It's taken several years, more or less, since episode 1 of TWiV to get here. However, 99% of what you say I now understand, including gel electrophoresis, Western blot, 16S subunits, etc., etc. This makes listening to TWiV much less infuriating.
Less infuriating or not infuriating?
Less.
Too bad. It's still infuriating.
Yeah, as now I rarely have to look things up, except for things relating to the papers you discuss. I guess I should give some details of my background. I studied electrical and electronic engineering. I'm an engineer working on water and wastewater treatment.
Nice.
In doing this, I've done some work in laboratories testing samples of water. I've also spent many an hour staring down a microscope, identifying the various types of bacteria and protozoa that actually do the work of treating sewage. Not to mention trying to identify and find ways to make life difficult for foam-forming filamentous bacteria. If you can imagine a 1.5 million gallon aeration tank at sewage works with such foam, then a strong wind blowing bus-sized chunks of foam about, well, you get some idea of my feelings towards filamentous bacteria.
Thank you for that image.
Yes.
Bus-sized chunks.
Bus-sized chunks of sewage foam.
Yum.
So I wasn't
Schmutzdecke.
There you go, Schmutzdecke.
Schmutzdecke.
So I wasn't unaware of biology, but treated it like a car. I could, in effect, check the oil, water, and simple stuff. Anything heavy duty I would leave to the lab. I hope you don't mind me blaming you, as it were. I think this is an interesting topic, and I'm enjoying learning about it. I guess that was your intention, to spread interest in a topic you clearly love. If I get the chance, then I will be taking some sort of biology course with the view to ending up in virology. Though I'm quite settled in my current career, who knows what opportunities the future might hold. I guess you can file this particular listener under the heading Mission Accomplished. Thanks for taking the time to produce the podcast that you do. I thought I should pass on my story so you can see just how good a job you are doing. P.S. Please excuse the brevity of the email, but I typed it on my phone.
Not brief.
P.P.S. I think I have removed all the autocorrect blunders, but if any got through, I can only hope they are funny. All right. And now, thank you very much, Richard. That's pretty cool. I like that.
Yeah,
very much.
that's awesome.
Alan, I think you should read the next one.
Yes, so the final letter for this show. Renato writes, Dear Professors Racaniello et al., thank you for the TWiV, TWiP, and TWiM podcasts. I find them immensely interesting, even though my wife forbids me to listen to them during meals. Well,
I wonder why.
Bus-sized foam things coming on your... Pass the foam, darling.
Her loss, really.
That's right.
I'm a computer science graduate student at Universidade de Sao Paulo, Brazil, and would like to make a suggestion to you. Recently, you discussed an episode of a podcast named Futures in Biotech, which I listen to regularly, along with some other podcasts, which are syndicated in twit.tv. That's This Week in Tech, major podcast syndication network. They have a very interesting podcast about computer security called Security Now. It says, hey, that's my pick of the week, a show about viruses that make you poor. That link. The host of Security Now does something which I believe you should also do. He pays to have his podcast transcribed and then publishes the transcription online. By doing this, the contents of his shows can be indexed by Google and other search engines. If your podcasts also had this, I believe many people would discover you by accident by simply Googling virus, parasite, and microbial-related words. Again, thank you all. Cheers, Renato.
Great.
Yeah. Transcripts are a good idea. They're a bit expensive. I've looked into it, and I don't think we could afford it. Not right now.
Transcripts with technical terminology are particularly pricey.
Someday, maybe. But it's a good idea. It would help with indexing.
Yeah.
I understand Google is working on indexing audio.
Really?
Wow.
Cool.
Yeah, I'm sure they could do it. All right. Let us make some picks of the week.
Excellent.
And Mark, do you want to have a pick, or do you want to pass?
I actually have one, and it's already in the doc.
All right, why don't you go for it? Tell us about it.
Okay, this is the guest pick of the week. So my pick is two guys in England who call themselves the Slow-Mo Guys.
This is really great. I didn't know this was you. I looked at these videos. These are great.
Yeah, and they have a YouTube channel, which I put the link to. And what they do is basically they have some pretty nice video equipment that takes very high frame rate, high resolution videos, and they just do fun things.
It's great.
Some of which, and they record them, and some of these can more or less be categorized as scientific. And one I provided a link to is an example where they recorded a dragonfly eating a little fly in flight in slow motion, and that's a good video. And I think the nice thing about these guys is in their videos is it captures, what can be sort of the fun spirit of investigation and discovery, which I think is one of the main reasons to do science.
Absolutely.
This giant water balloon video has had 19 million hits.
Yeah, these guys are viral. My oldest son showed me this some time ago. He was just so turned on by this.
There was a TV show on the Discovery Channel that they were on, or something similar to that was on. showing bullets going through like watermelons and stuff.
Apparently they work for a company that has these high frame rate.
Yeah.
I mean, huge frame rate, hundreds of thousands of frames per second.
That's right.
Wow.
Amazing.
It is.
Cool. Thanks,
it is.
Mm-hmm. Kathy, what do you have?
So I have a pic that I want to give a little background on because we do some things with mouse genome. I get an automatic feed from the University of California, Santa Cruz, UCSC, genome browser whenever a new genome is published or uploaded. And I thought this related a lot to some of the discussions that the postdocs at ASV were having, and also came to the front on today's show, where, knowing some things about animals and more about animal viruses is a worthwhile thing. And also that we'd like to have a way to convey or have the public have more of an appreciation for basic research. And so this link is that the genome sequence of the medium ground finch, which is one of Darwin's Galapagos finches, has just been released.
Excellent.
And this is the first of a collaborative process between the BGI, Beijing Genomics Institute, and the Genome 10K project. So the link is to the press release, and one of the comments is that it's I'm not finding it here. Anyway, that it's important both from a biological standpoint and for historical significance, and so on. And if Mark and Joe hadn't had the boa constrictor genome sequence, they wouldn't have been as easily able to subtract that out and get their virus sequence. Yeah. So I think the more genome sequences that we can know, the better. The 10K project that I mentioned is to sequence 10,000 vertebrate genomes, representing approximately one for every vertebrate genus. So
wow,
cool.
Yeah,
great.
Alan, what do you have for us?
Well, my pick is on the CDC website. They have just finished scanning and putting online the first 30 years of Morbidity and Mortality Weekly Report.
Oh.
So now this archive, I guess they must have started computerized typesetting around 1981, which is when their archive used to go back to. Now it goes all the way back to when this particular journal started, 1952. So you can go all the way to number 1 of MMWR, which started out as almost kind of a newsletter and has, of course, grown into one of the top public health journals in the world.
Oh, here's one from January 2nd, 1953.
Goodness.
It's my birthday. It's my birthday.
Ah.
And what would they be concerned with then, do you think?
Leptospirosis, diphtheria,
Polio?
salmonella.
Polio?
Yeah, polio.
Hepatitis, gastritis. They don't even mention polio.
What?
Not in the summary.
Come on, I was 13 years old.
Maybe it wasn't notifiable in 1953.
Had to. Oh, my goodness.
It had to
Maybe
be.
they
I
didn't know
agree,
what it was.
but it is not here.
Gee whiz.
Good point, Dickson.
Wow.
I'll have to. It's like having a vintage from your birth year,
That's right.
Now I have the Morbidity and Mortality Weekly Report.
I have a great landmark for mine. It was the onset of World War II.
Let's see if I can guess. 1939?
1940.
Really?
Yeah,
I thought it was 39.
Well, depends on which arena.
Okay.
Yeah.
No pun intended here.
Rich Condit.
I'm going to squeeze in real quickly 2 picks because they're episode relevant. One is an article in the Gainesville Sun, our local newspaper, that as I look at this, it looks like it showed up in the a bunch of different papers that describes the recent capture of a giant python out of the Everglades.
Sure.
This thing was 17 feet long and had 87 eggs in it.
Exactly.
Yeah.
So as I briefly mentioned before, there's a problem in Florida because pythons that have been released from people who had them as pets and they got too big have inhabited the Florida Everglades and now it's not clear. The estimates are between 5 and 180,000. and they're wiping out whole species of vertebrates in the Everglades. So this is a problem, and this is well, I guess it's a problem unless you're a python.
It's a tremendous success if you're a python.
That's right. It's interesting. So that's a pretty big snake they caught. And the other is I thought I would link the California Academy of Sciences.
Nice.
This is a wonderful museum that I visited, and they have the Steinhardt Aquarium, which itself is amazing. And their, as Mark I'm sure will attest, their herpetological collection is remarkable as well. So anybody who's in the area, stop and have a visit to the California Academy of Sciences. It's wonderful.
San Francisco, right?
Yep, that's right. In Golden Gate Park.
I'm going to be in San Francisco in September. I'll go.
Is the Fly Shacker Zoo still open?
Which?
The Fly Shacker Zoo. That used to be the zoo of San Francisco.
Yep, that's still there also.
I was raised in the San Francisco area as a kid, and I used to go there.
So in the process of this project, Mark, did you visit the Academy?
Oh, yeah. They're actually, we collaborate with them on a number of projects. And
so have you run into a guy named Bob Drews?
I didn't, no.
Okay. He was their herpetologist for a long time. I think he's retired now, or at least semi-retired.
Mm-hmm.
Okay.
This is a picture. I'm looking here at the BBC NPR story. It's a picture of Taryn with her boa. See that, Dickson?
I do see it from here, yes.
To this, it's pretty cool.
I see it.
Mark, so you had some sick pythons, but you didn't get virus from them, is that correct?
We did. They were sick, but they were not diagnosed with IBD. So they were samples we got from the pathologist, but they had been tested for IBD, or they've been examined by histopathology for IBD, and they were negative. So we haven't been able to get any IBD positive python samples, which we would obviously love to get our hands on.
But you got the arena from those samples or not?
No, we have only gotten the arenavirus from boa constrictors and
okay.
annulated tree boas.
And then you had another partial sequence, I think, right?
Yeah, that's from a boa constrictor. And actually, since we published the paper, we've been collecting more samples, and we've expanded the number of viruses a little bit.
Nice.
All from boa constrictors.
Okay. Who's next? Dickson, do you want to make a pick?
I do, actually. I would be neglect if I didn't try to inject some non-virological humor into this. So this is a poem by Shel Silverstein. He used to be the poet in residence for Playboy magazine, but he also has written some wonderful children's stories. And the poem that I'm going to refer to is called I'm Being Swallowed by a boa constrictor.
Very good. Yes.
And you know this poem, right? Because it's an incomplete.
Peter, Paul, and Mary sang it as a song.
It's an unfinished symphony. It ends up, with the lines, "I'm being swallowed by a ...
Because he was swallowed, right?
He certainly was.
Cool.
So just as a way of keeping track of all this.
Good thinking, Dickson.
Thank you.
Yeah,
I knew there was a reason we kept you. I'm glad you laughed at that.
I did.
All right, my pick is a very nice video made to showcase the Department of Microbiology at Mount Sinai.
Uh-huh.
And it talks all about the research that's going on there. And since I'm a graduate of that program, I'm promoting it.
Is this put together by Peter Pilates?
No, it's put together by, I don't know, it looks very professional. But anyway, if you go to the webpage, the first guy you're going to see there is Matt Evans.
All right.
There, and they go through a bunch of their faculty, who are mostly virologists, and they talk about what they do. But it's pretty cool, and anybody looking to go to a PhD program and do virology, just check it out. And that's it for us. We have a couple of listener picks. We had Renato, who gave his pick, which I've already forgotten what that was. Oh, that was the virus guy. Yeah, the twit.tv podcast. We have a link sent by Robin. And this is a way to get the weather. You enter your city and state, AccuWeather.com. Why would people think we're interested in the weather? I have no idea.
I have no idea.
Fresno, California. I don't know why it's got Fresno in it. Maybe that's where he's from. It's 105 degrees currently.
Yeah.
I remember driving through there. It got really hot. Yeah.
Yeah.
Oh, New York. Let's see. Yeah, 86 degrees. It's in Fahrenheit. There must be a way to change this. All right. Well, anyway, so that's from Robin. And we have from Chad, one of my Facebook friends.
The country or the person?
Person.
The person.
Chad said, one of my Facebook friends posted this Science Daily article. Thought it might make a interesting listener pick. Listen to this, Dickson. Mm-hmm. Rare protozoan from sludge in a Norwegian lake does not fit on the main branches of the tree of life.
I don't believe that.
I don't either.
Well, then make another branch.
It's probably Schmutzdecke.
Yeah.
No,
maybe
I don't think we
they
know all the branches of the tree yet.
There may be sequences that are just unknown.
I saw this press release. So Science Daily is a site that just posts press releases.
Right. It does not make a judgment.
It's not. They don't do original journalism. And I remember seeing this thing and thinking, what is this even talking about? So one of the lines in this press release, they saw the protozoan did not fit in on any of the main branches of the tree of life. It's not a fungus, alga, parasite, plant, or animal.
Right.
That's not how I learned taxonomy.
Why did they call it a protozoan then?
Yes.
So sometimes Science Daily has a link to an article at the end, but this one says that this article is based on materials provided by the University of Oslo.
Yeah.
And if you click on that, you just get another press release. So I don't know about that. I've heard of things that don't fit on trees of life, and they always turn out to be just unknown sequences.
Yeah, dark matter.
Dark matter.
The tardigrades are a good example
Yeah.
of a branch of life that's got rare forms in it, but that's a branch. You just make another branch if you're stuck for what to do.
Hey, Mark, what do you think about sequences that you can't classify?
I think that we see them all the time, and it's hard to know what to make of them. I mean, sometimes you sequence stuff and it's just sequencing error, so it's just phantom sequence.
Yeah.
It's not even real. It doesn't derive from a real nucleic acid.
You mean the machine is just twiddling its thumbs?
Yeah, it's just spitting out,
just practicing
stuff. Yeah.
As Eric Delwart said, DNA is really a problem. It's everywhere.
Yeah, that's right.
Yeah,
that's right.
I was just we just discussed Forrest Rower's work on TWiV last week. Do you know Forrest Rower, Mark?
I've not met him personally, no, but I'm familiar
yeah.
with his work.
So he estimates that there are 2.5 billion viral genes out there that have yet to be discovered.
Yeah. So that's plenty of work for people to do.
I think we should get them all. I really think we need to know the whole.
Eventually, we will. That's our charge in life.
Not if NIH continues in its current path. I was just at a meeting and someone told me that a program officer from NIH told him that NIH does not like R01s, investigator-initiated grants, because they can't control what goes on. Can you believe this? NIH wants top-down control of research.
No,
well,
I'm not buying it.
I don't think that's a universal feeling at NIH.
This is from a program person, not
one program person.
It's an institution that's ultimately full of scientists.
Not even a program person. It's the head of one of those branches, which I don't want to name because then everyone will know. It's quite a high up person.
Yeah, but I'm sure the administrators do feel that way, but it's just that's not the point. That's not what they're there for.
No.
Well, if it's anywhere near that, I don't like it because it should be. You can have a little bit of translational stuff, but
Vincent, if we're on study section and we give a grant a 1, what are they going to do? Right? They got to fund it.
That's right.
All right. So that's bottom up. We can have an impact.
Yeah, I think that's important to remember, everybody.
Yep.
And on that word, we will wrap up this TWiV. And I now know that I can handle 1, 2, 3, 4, 5 Skype connections here on the university internet. Pretty good.
Amazing.
I bet.
TWiV.tv is where you will find TWiV. Also, we're on iTunes. And if you're new and you like us, subscribe and leave a comment or a star over at TWiV. It helps us to keep visible, and that lets more people discover us. And you can also find us on Facebook, facebook.com/thisweekinvirology. And finally, of course, Dickson, what should we tell people to send us?
Send us their Pics of the week.
Okay. Or they can send us their questions and comments.
Of course.
To TWiV@twiv.tv. I want to thank everybody for joining us today. Joe DeRisi, of course, who has left. He's at the University of California, San Francisco. What department is that? Biochemistry and biophysics, Mark?
That's right.
And thank you as well for joining us on TWiV, Mark. Hope you liked it.
Yeah, it was quite a pleasure. Thank you for having me.
Yeah, we had a lot of fun with you.
Thank you, Mark. This has been good fun.
That's great.
It has.
Mark, you said at the beginning this might be my best shot, but I'm sure you'll be back on TWiV.
All right, we'll hope so.
Just keep keep the passion, man.
All right.
Well, if you become an NIH program officer, you're not coming on TWiV.
Unless you say the right stuff.
Well, yeah.
He may say I love R01s.
That's right.
There you go.
Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks. It was a lot of fun.
You have a good connection there. I like it. Something about that Ann Arbor thing. I don't know.
Okay.
Rich Condit, University of Florida, Gainesville. Thank you, Rich.
Yeah, pleasure as always. Good fun.
Sorry about those pythons.
It's okay. But, I have a thing about this. Who decides what's, an invasive species and what is the endogenous stuff? And there's judgments going on here that I'm not real sure of. So the pythons are taking over. That's going to be temporary, and they're having a good time. That's okay.
There is actually quite a lot of debate about that in the ecological literature. I think, right
there
now.
is. In fact, we are an invader for this concept.
Yeah.
We are too.
Yeah.
Sure.
That's right. I spent 6 months in Australia, and they were trying to get rid of the brown trout, which was brought in from Europe. And I said, the first thing you should do is get rid of the thing that brought the brown trout to Australia, namely the Australians. And they didn't look up at that. They didn't like it.
Alan Dove is at alandove.com. Thank you, Alan.
Always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.